Okay, so it is now 3 o'clock Eastern. So I'm going to get started just to make sure that we stay on track. This meeting will be recorded. So if at any point you have to step away or if you miss any part of it, please feel free to watch the recording. So welcome everyone, and thank you for joining this important webinar to help you prepare for meaningful participation at the ELPFDD meeting on kidney xenotransplantation. This meeting is taking place on November 9th in Bethesda, Maryland, and today's webinar will provide an overview of kidney xenotransplantation and the considerations of risks and benefits. Today's webinar is led by our ELPFDD chairs, Drs. Vanita Kumar and Dr. Peter Rees. So for more information and to register for November's meeting, please visit our meeting webpage at www.kidney.org/EL-PFDD-Xeno. You can also use the QR code located on this slide. We have saved the last 10 minutes of today's webinar for questions. If you have additional questions or comments, please email me directly after the meeting at heather.murphy@kidney.org. So before we jump into the presentations, I wanted to say thank you to our meeting sponsors, eGenesis and Makana Therapeutics, who have provided educational grants to support this important initiative. So our first speaker is going to be Dr. Vanita Kumar. She is a transplant nephrologist and director of the high-risk transplant program and has involved in the xenotransplantation efforts at the University of Alabama in Birmingham. Dr. Kumar will provide an overview of xenotransplantation. Welcome, Dr. Kumar.
Thank you, Heather, and good afternoon, everybody. Thank you for being here. I'm looking forward to sharing a little bit of information and then engaging in a conversation with you. I'll get us started for about 20 minutes, and then Dr. Rees, my partner, will jump in. And after that, we look forward to communing with you. So with that in mind, let's get started. I am going to, over the next 20 minutes, present to you my journey in xenotransplantation of what led me to go from xenotransplantation, are you nuts, to actually believing it and engaging in it. And we'll do it through these particular objectives. You'll have access to these slides, so I won't read the objectives. But I want to first start off by talking about why even xenotransplantation. Most of you on this call are probably familiar with this fact that in 2023, kidney transplantation is an amazing life-saving option. We would want that option to be available to all our patients. And yet, of the 90,000 patients that were looked at during this time period that were waiting for a kidney transplant, representing a fraction of people that actually have end-stage renal disease, which is more than 500,000, only 25,000 a year got a transplant. In the last couple of years, that line at the top, which is the number of people waiting, versus these bars at the bottom, which are the number of transplants that have happened over time, the number of transplants have gone up a little bit, and that line has gone down a little bit towards the end, sadly, because of COVID pandemic and losing a lot of dialysis patients. So it's not for a good reason. This big gap continues to be there. Unless we think this is a US-centric phenomena, worldwide, there are about 10 million people that are waiting for that are undergoing renal replacement therapy, and at least half to a quarter of them are transplant recipients. And while they wait to get a transplant, they're undergoing peritoneal or hemodialysis. And just these facts here are pretty sobering, that if you think about some of the common cancers that we worry about, prostate, breast, bladder, and colon cancer, the 5-year survival for all of those cancers is better than the 5-year survival for our patients who are undergoing peritoneal or hemodialysis, such that the survival is about 42% at 5 years. Most of our patients then, unfortunately, are dying waiting for a transplant within 5 years, and fewer than 1 in 7 patients with end-stage kidney disease actually make it to the waiting list. So the problem is huge. Organ transplantation is life-saving. Where are the organs? Do we have sufficient organs? And for that is why scientists have been looking towards other ways of enhancing and minimizing this difference, one of them being xenotransplantation. And for a long time, it was called the impossible solution. So let's step back a little bit and take a historic look at xenotransplantation. Now, it's an ancient notion of combining animal and human parts, the creature of Crete or Medusa with the snake head, or where I grew up in India, in my younger years, Lord Ganesha, where it's the head of an elephant on the body of a human. So there has been Mesopotamian and other cultures that have had this notion, but the first recorded solid organ transplant in modern medicine goes back to Dr. Gerboulet from France, when he encountered a woman who was 48 years of age, who had acute kidney failure, was going to die, and he was compelled to do something about it. And after looking around, he said, what if I used a pig as a source of an organ for her? He actually transplanted her. The organ lasted 3 days, unfortunately clotted. He went home thinking he's a terrible surgeon. Little did he know that he actually was the mentor to Alexis Carrel, who got the Nobel Prize for vascular surgery as in hooking up vessels with vessels. And if that happened in 1906, when he first tried his experiment, initially with a pig and then a goat kidney, eventually over the years, up until 1984, other forms of animals were used. This time, macaw, a chimpanzee in 1963, where a 23-year-old woman had 9-month kidney function. 1984, where a baby born with a congenital heart disease had a baboon heart. Baboons, chimpanzees, macaws put together, they're called non-human primates, but non-human, we're primates too. So they're non-human primates. That's a term you may come across. And so if today we used a non-human primate, we could probably do better. So why did we stop? Why are we thinking about pigs as potential organs? And that's because, one, there are not enough non-human primates to make up for the demand, and two, the idea that they may harbor viruses that may be bad for humans. Actually, the FDA in 1999 banned the use of non-human primates for use of xenotransplantation in 1999. So scientists turned then to other sources of animals, and pigs became the preferred source because of these reasons. They're available in plenty. You can actually engineer and change the DNA of the pig in such a way that it reduces the risk of rejection, because the humans behave differently against that. That the pigs are sized similar to humans, and they're thought to have limited risk of transferring infectious agents. Also, pig source is already in use in medicine. Just some examples: when you have a burn, a live pig skin is used to temporarily close the burn wound. Pig valves have been in use for humans for over 40 years, and corneal transplants are other things that are used. And so because of all of these reasons is why pigs were thought to be a good source. So what did then scientists do with this particular source? What were some early experiments done, and barriers overcome? So the first thing that the scientists came up with is a way to study this before it gets to humans. How do we study if pig kidneys will work in humans? They said, let's take a non-human primate, a baboon, and take a pig and transplant it into the baboon. So if you took a donor pig that was not modified, you didn't do anything, we call it wild type, and you put it into a baboon, after you do the surgery, the kidney work looks great. But within minutes, this is what happens to the kidney. And they did this experiment multiple times, and the kidneys would turn black, would not function. This is called hyperacute rejection. We dread this happening in transplantation. And we know this happens because of some form of antibodies that we have against organs. So if you take a human to human organ, and if you don't match them right, either blood type or even HLA type, and the human has a very strong antibody to this other human organ kidney, this can happen. We have technology now that prevents us to see hyperacute rejection in humans. So the idea that when you put a pig kidney into a baboon, this happened, they thought, there's something in the baboon that's causing the pig kidney to be rejected. And sure enough, we found that baboons have naturally occurring antibodies to pig organs, and humans and baboons both have them. And in the initial experiments, what they then started doing is heavily treat the baboon, the recipient, with immunosuppression to try and reduce this rejection risk. And they ended up really over treating the recipient. So they found out what was causing the rejection. They actually found 2 or 3 different antigens. These are naturally occurring proteins on the pig kidneys that humans and baboons have antibodies to. So they found multiple different ones that we were reacting to, but they didn't know how to sufficiently reduce the antibody load in the baboons to allow for function of these kidneys. Along the same time, technology was evolving in other fronts. Cloning or gene editing came into play, where you could then change a portion of an organism's DNA. And this then got the scientists to have a brilliant idea. I call it the Eureka moment. The Eureka moment was modify the donor. We are doing all these things to treat the recipient and to treat the recipient antibodies and immune suppressing the heck out of them. Why don't we just take the donor pig, and modify it in such a way that it looks more like a human? Brilliant. And so what did they do? These antigens that they found that the baboons or humans have against pig, you can actually change those in the pig and knock them out, remove them. You can also then add in genes into the pig that make the pig look more human-like. Wow, now you have an organ, a potential organ that looks more human-like, that then could have better outcome. But you still needed to study this. So if you look at a historic perspective from 1906, 100 years of work, what happened in those first 100 years of work? Well, in 1994, these antibodies were discovered. And then 2004 is when the cloning technologies became available. And then what happened between 2004 and now? Okay, what happened was, now instead of using the wild type pig, these modified donor pig organs were used in the baboon model. And what do we learn when we did that? Well, one, we learned that modified pig kidneys, the knockout kidneys, the genetically engineered kidneys, whatever term we want to use, they work pretty similar to human kidneys, doing all of these functions outlined here. Were there differences? Yes, there were differences. Our human kidneys make hormones that work on our bone marrow to make red blood cells. The pig kidney also makes the hormone. It's called EPO. For those of you with kidney failure, have probably received this hormone and on dialysis or in your organ transplant phase. The pig kidney makes the hormone, but the human bone marrow, or in this case, actually the baboon bone marrow, didn't work. And so the baboons needed continuous EPO replacement. Well, what about the pig EPO in humans? Will that work, or will humans need to take EPO long term? Something to consider. The pig kidneys continued to grow in the baboons because they were taken from the pig when the pig was 6 months old. And so the growth continued, something else to think about. Also, an interesting thing is rejections continued to happen. Now we were able to continue to have the baboon keep the pig kidney for initially days, then weeks, then months, and some went out to a year. However, later there was still rejection being found. So what do we do? Why is this happening? What else can happen besides the gene edits? So as the immunosuppression was being applied, again, they started to use immunosuppression that was not necessarily standard immunosuppression. One of the breakthrough thoughts, again, was, look, we're making these pig kidneys to look more like humans, but we're putting the pig kidneys in baboons. We're not And the pig kidneys are not being made to look like baboons. They're being made to look like humans. So we have to try them in humans to know if this is working or not. Well, how do we take something that is still in this phase and just directly put it into our humans? Because we need to know if this is going to work or not, or more importantly, if this is going to cause harm. Is there a way to figure this out? Voila, another brilliant breakthrough, thinking about this brain antecedent model, where if you took somebody who is past, who wants to be an organ donor, cannot be an organ donor for whatever reason, they're brain dead, can we now ask permission from their family to use these modified pig kidneys in them to test our hypotheses that these pig kidneys are made to look like humans but are being studied in baboons? What would the human body response be? And that's where this last couple of years we've had additional progress made. So what were some recent events and questions answered? One, the xeno kidney transplant surgical process, so euthanizing the pig, versus taking the organs from our organ donors, deceased donors, or even living donor. Similar surgical process, similar way to transport them, similar way to do the surgery and hook it up into usually the groin, just above the groin on the right or the left. That's typically how we do it in humans. You can do it here. This is a picture of where we were actually able to get a pulse of the donor kidney. This was a beautiful gift we could give to our donor family, where we recorded this, put it into a plush animal toy, and gave it to our recipient family. I'm sorry, our decedent's family, who to this day tell us how meaningful it is, because they can hear the pig heartbeat, so the pulsation of the pig organ in their loved one. We also did this experiment using standard immunosuppression with us that we use for the time of transplantation. And the New York experiment had also the standard immunosuppression, and we can talk more details about that. But there was no rejection. More recently, we just published this paper where we talked about 7 days out, there was function, and the pig kidney cleaned blood just like it would in the human, and actually made urine like a living donor human would. In the first 24 hours made 37 liters of urine. And sometimes our living donor transplants, when you put it in our recipients, they make volumes and liters of urine. Gallons is what you would say, eventually settling out to about 5 liters. In the short time period, there was no pig to human disease transmission. So these were some of the advances that the recent experiments were able to show us. And I show you this slide not to have you get lost in the information, but just to say in 1985, when the first pig to baboon model was conceived, it was in the last 2 years that we moved from that to the decedent model, applying all the knowledge learned before then and since then to come to where we are today. And let's contrast this with human allotransplantation. If our first kidney transplant and solid organ transplant kidneys led the way. So the first transplant happened in 1954, where a twin gave a kidney to another twin. It wasn't, and here they were both genetically matched from DNA point of view, because they were identical twins, and a chromosomal match, right? And that's why they did not need to use immunosuppression. But when they had to progress from there to using organ transplants between unidentical twins, or even siblings who were not twins and not identical, to eventually going to deceased donor organs, that transition didn't happen overnight, took time, and continues to happen. And the history of allotransplantation is all about refining the surgical technique and refining the immunosuppression. And these early advancements came from a lot of practice, along with simultaneously building our ethical and medical consensus on brain death and consent. So over 100 years of xenotransplantation, from the first experiment from Dr. Jubilee in 1906 to now, we have had multiple kidney transplants in decedents and now 2 heart transplants in humans. Many questions have been answered. So are there remaining barriers? Are there additional knowledge gaps? What are the next steps? Where do we go from here? At this point, I turn this over to my wonderful colleague, Dr. Rees, who will help us really think through some of these contextual things before we get to our Q&A. Thank you, everybody.
Wonderful. I'm just going to share my screen.
Thank you, Dr. Rees. And just to give you a brief introduction, our next speaker is Dr. Peter Rees. He's a transplant nephrologist and ethicist at the University of Pennsylvania in Philadelphia. Dr. Rees will review the benefits and risks of kidney xenotransplantation. Thank you, Dr.
Thank you very much. It's an honor to follow that great presentation by Dr. Kumar. And what I'm going to do is sort of synthesize it and think a little bit about how I would talk to a patient about participating in a xenotransplant study and who I might think would be appropriate and what I would want them to feel comfortable that they understood. So I just want to give a little bit of just brief disclosure that I do have a grant from one of the companies developing xenotransplant technology. It's not commercialized yet. So what I'm going to talk about is the longstanding ethical problems that have been with us for 50 years of talking about xenotransplant, the known risks, the unknown risks, how we would think about patient selection, and how we would counsel patients. I'll go through those one by one. So the ethical problems related to xenotransplant are things that were recognized, many decades ago, and really they haven't changed very much. I think what's changed a little bit is the technology that can help to address them has improved. So I think the first one is that anyone who is thinking about participating in a trial where they would have an animal organ transplanted into them, say a kidney or a heart, there's going to be a risk of organ dysfunction for that recipient. It's going to be hard to know whether or not the risk of rejection is going to be 5% or 20%. There's going to be a risk of that organ being lost, and those risks are because that human body's immune system is being exposed to antigens that are not human. And the immune system interacts a lot with the blood clotting system, so it's also possible that the person might experience problems outside that organ with blood clots or bleeding. There would be a risk of infection to recipients and to bystanders, like family members. There's a risk of a loss of privacy, and I think we live in a different world than we used to with social media, everyone carrying phones around. The spread of information is faster than ever, and there is a possibility that someone who participated in one of these trials, that their name or the names of their family members, the city they live in, could become public, even though these things by law should remain private. And as a result, I think that it leads to some complicated decisions about patient selection, consent, education, and who needs to be consented, who needs to provide consent. So what I'm not going to talk about in terms of ethics is I'm not going to talk about animal rights. I'm not going to talk about natural law. Those would be topics for a different day. So we have seen in the media and in scientific journals some reports about xenotransplants, sometimes inside a study context, sometimes under what we might think of as emergency authorization. And the experimental data are pretty limited, but we can ask ourselves, what did we learn? So as Dr. Kumar pointed out, hyperacute rejection, which we have seen in the past, and this was avoided. So when the kidneys or the heart were put into the bodies, into these human bodies, those human immune systems did not reject the organs immediately. But several studies so far have shown suggestive evidence that there was rejection emerging in these experiments. So I'll give you an example. In the Maryland heart transplant, it failed after 2 months, and the failure of this organ took place for reasons that I don't think that anyone feels confident to say that they know why. But if they looked at the organ tissue when it was put in, it looked very healthy. And when they looked at the tissue when it was taken out, it looked very different. It looked very unhealthy. And the possibility that rejection caused these changes in the tissue and caused these this dysfunction of the heart tissue it's very possible that that was the explanation. And then, as you may have read, that there were also studies and reports of kidney transplants from New York University. 2 brain-dead individuals were brought into New York University. They received pig organs, and those pig organs did filter the blood. But when they did biopsies, and they did very careful studies of these biopsies, and then they looked at kind of molecular analyses with special immune staining and gene expression, it looked as though there was emergent rejection going on in those kidneys as well. So the kidneys were filtering the blood, they were making urine, and it did appear as though they were removing byproducts from the blood. But when they looked very carefully at the kidney tissue also seemed to change in its appearance over that time period. So putting this all together, I think that the risk of rejection remains substantial. It's hard to say how big it is. And I think as these additional studies go forward using brain-dead research donors or studies under emergency authorization with heart transplant, that there's going to be a rapid learning curve. There's a lot to know in terms of trying to safely determine the compatibility of any given patient with an organ. We have a lot of methods to do this, but those methods have to be adapted to this setting. And the best approaches to preventing and treating rejection are not known yet. There are many available therapies, some of them experimental, that they could address rejection, they could prevent it, they could treat it, but those therapies also carry meaningful risks. And what those risks are is that if you lower a person's immune system's ability to function to accept the organ, then you increase the risk that infections or cancers in the future could emerge. So we know that in our general practice with human donated organs, that we have to immunosuppress the patients, and they can, in some occasions, become very ill from viruses like cytomegalovirus, and may or may not be one you've heard of, or even common bacterial infections such as a urinary tract infection. And some of our patients, unfortunately, can also develop lymphomas. So these are issues where the patient develops infections as a side effect of the immunosuppression that they receive. And we would expect that that could be an issue here. It might even be more of an issue if a lot of rounds of treatment with immunosuppression are necessary to suppress rejection. And then in addition, we also have to ask ourselves, what did we learn about infection risk in terms of organisms that live in the pig being transmitted to the recipients? So one thing that did appear to take place is that a pig virus called porcine CMV does appear to have been transmitted from the pig donor to the human recipient in the first Maryland heart transplant. It's unclear when that pig virus was transmitted to the human recipient, whether or not that virus was causing damage, whether it was replicating, but this transmission did appear to happen. And there may be solutions to prevent porcine pig CMV from being transmitted. There are ways to quarantine the herd of pigs. There are ways to screen the pig probably more rigorously than it was screened in that first heart transplant case. So in principle, we should be able to bring that risk down quite a bit. And then as Dr. Kumar also, I think, alluded to, sometimes the genome of animals or humans can contain retroviruses. These retroviruses live in the genome, in the genes of a cell that comes over, say with a pig kidney or a pig heart. And it is possible to remove some of those retroviruses from the pig genome. And some companies that are developing and breeding pigs for organ transplant have been able to remove some of those viruses. And there may be other infections that pigs have that should be able to be identified in the pig and prevent transmission to the human recipient. So my summary is that transmission of infection from the pig to the human in organ transplant remains an issue that we need to worry about. But I think perhaps less so than in rejection, we should be able to reduce this risk to a very low level, close to zero. So what do we learn about privacy risks from these recent transplants? Well, there have been very many, so we don't know. I'll narrate what I think happened with the Maryland heart transplant, which is that the University of Maryland, of course, is bound by all the same rules that all the hospitals are, that patients' names and identifying information must be kept private. But somehow, this person's identity became known, and the world learned about this first Maryland heart transplant in many ways through press releases and lay media reports. There was kind of a narrative, in a sense, that was carried forward about surgeon as a pioneering hero. And we always have to be a little bit worried about that because these studies are done by large teams of people. There are many different people contributing, and I don't really think of any of them as either heroes or villains. I think of them as scientific clinical teams trying to do hopefully what's best for the patient. The name of the patient became public. And then something I think very disappointing and even weird happened, where the Washington Post discovered that the patient had a history of a violent felony crime. And then they promoted this sort of naive and bizarre narrative that he might have been undeserving of transplant, which was totally tangential to what was going on and probably irrelevant. But that drew a lot of media attention. And so I think the implication is that hospitals may, despite very sophisticated efforts, they may be unable to guard the patient's privacy. After all, hundreds of people might participate in a case like this. If we think of everyone from patient transport all the way to the surgeons who do the surgery. So medical privacy may be hard to protect, and the narrative may be hard to control. So what are the implications for families and close contacts? Well, I would say that xenotransplant requires a different conceptual model than a lot of studies that patients might consent to, because the conceptual model really has to account for theoretical infection risks that aren't just necessarily an issue for the family. They could spread beyond the family to a close contact in theory. And so the people who need to be educated about these risks are not just the patient, as is the case in many studies, but their family members also need to know that in a way they're going to be involved in this study. And really that's true for medical teams as well, but at least medical teams probably are used to being in a research environment. So the caregivers are going to have needs. How to meet the needs of these caregivers is not something that I think we know enough about. And there'll have to be education about the burdens to the patient, the family, the caregivers, not just related to infection, but also to their privacy. And hospital staff will really need that education too. So we're used to, in research and in talking to our patients, thinking about what are the needs of the patient. In this case, the needs and to some extent the risks are broader. They include that patient's close community. So I think I've emphasized to you that this is an experimental therapy. And so you might be wondering whether or not Dr. Kumar or I could imagine encouraging or counseling a patient about participating in a xenokidney trial. And if we could imagine it, who would we have in mind? So I'm going to tell you who I have in mind. And I'm not going to say that I have the right answer, but at least I have thought about it. So I would be willing to talk to a patient about enrolling in a xenotransplant trial based on what I know. But my personal view is that it would not be for everyone. I would be willing to talk to a patient with a very specific set of circumstances, not the average patient who I meet in clinic. So the profile to me of an acceptable patient would be someone who is very disadvantaged by the organ transplant system. So they don't have a really feasible, imaginable path to transplant in the near future. Certainly, they don't have a living donor who might have come forward. Certainly, it would not be a person who has many years of waiting time and is getting kidney offers already. I think it would be a patient who's much more likely to die than get a kidney transplant. However, I think it also has to be someone whose health status is sufficient to endure a transplant. I don't think that I would accept someone for xenotransplant who had such severe heart disease or such severe diabetic ulcers in their feet that nobody wanted to take them to a human organ transplant because they wouldn't survive it. I think it would have to be a person with really, who had in one way or another demonstrated a lot of resilience in terms of their personal characteristics and social support, because it'd probably be hard to tell them with any accuracy what a xenotransplant might turn out to look like in terms of the treatments they'd need, the hospitalization days they might endure, the side effects ahead. And I think I would probably choose a patient who told me that their quality of life was very low, that they were very dissatisfied with the situation that they were in, for instance, on dialysis. I just want to let you know that we can predict with pretty good accuracy who are the patients who are going to wait many years for a kidney transplant and be unlikely to get one. We can predict with a pretty high degree of accuracy who are our patients who are more likely to die than get a kidney transplant. There's a tool that I worked on with Dr. Schold and some other colleagues called Titer, and we can sort of plug in some characteristics of the patient, their age, their blood type, how much waiting time they have already, and have a pretty objective measure of the probability that they're more likely to die than get a kidney transplant. And a tool like this could be used to try to choose patients who would be reasonable at a first step to enroll in a trial like this. So for instance, a patient between the ages of 50 and 59 with less than 2 months of pre-dialysis, pre-listing dialysis time, who has diabetes, we could meet them shortly after their time on the waiting list and talk to them about the fact that kidney transplant is probably not likely in their short term, and maybe they could consider other options. So even after sort of identifying patients along those lines, patients with a long predicted waiting time, I think we could also go to a second step and evaluate their overall state of health, their resiliency, and ask them, how unhappy are you with dialysis? Because for some patients, they just really haven't adapted to it. They really don't like it, and they're searching for something else. So if we were to then proceed and talk to a patient and say, what do you need to know? What do you need to demonstrate that you understand? So there's always going to be risks and benefits. And the benefit of a xenotransplant would be that they didn't have to wait anymore. They could get a transplant sooner, and it might work, and it might improve their quality of life. But I think that I would need to hear that patient say to me that they know that it is not possible to accurately estimate the risks and benefits. They would need to say to me that they understand that really it's an experimental therapy, and it's not possible to give them percentages to say, this is the likelihood that you'll be alive with a functioning transplant in 6 months or a year. I think they would need to say to me, Doc, I know that the kidney might not function well because it might develop rejection. And I think they would need to understand that the treatments for rejection might carry risks of infection or cancer or other side effects like high blood sugar, and that the kidney might not last long. I think that they would need to understand that there's a small risk of infection from the pig, and the main infections that we worry about are viruses, and that it's possible that an infection could be difficult to treat because the patients had so much immunosuppression. And this risk of infection transmission from the pig should be relatively small because of genetic engineering and the fact that the pigs are quarantined and tested, but it's not zero. And I think it would be important to know that even though this risk of infection from the pig should be really small, that if there were concerns, if the patient got a fever, and there were any worries that the patient could spread an infection to others, that the patient in close contacts might be required to quarantine. So I put this picture up here so that you'd kind of imagine it could be someone kind of stuck in their house. And if health authorities were worried that public health were at risk because the patient seemed to be infected and they were worried there was some chance it could have come from the pig, The patient might be quarantined, their caregiver might be quarantined, and they might not be allowed to withdraw from the research project. They might be forced to participate. They might be forced to give blood samples and things like that. So again, I think the risk of getting an infection that's a meaningful, dangerous infection from a pig transplant is very low. But if it did happen, the consequences could be fairly extreme. They might need to be quarantined, and maybe their family would be too. And the other thing that I would want them to know is I want them to know that if the kidney doesn't work, the patient presumably could return to dialysis, and it's possible the patient could get another kidney transplant later, but it's also possible that that door to transplant could permanently close for them. And why would that be? It could be that the side effects from the transplant and from the treatment were severe, that they became too frail. It's also possible that their immune system could be too sensitized to human antigens and that they wouldn't have compatibility with other human organ transplants. These are all possible. And I talked about the privacy risks as well. So I think in a way we have to look at this differently than we've been thinking about human organ transplant. We want, with education and consent of patients, we want emphasis that a lot is unknown and there's a real potential need for lifelong monitoring. We need to educate their close contacts, their caregivers, friends, and family members that there are these very small but non-zero risks of infection and its spread. I personally think that it would be a very distinct profile of patient who would be eligible for one of these trials. And I would need to know that they were very resilient, that they had strong social support, and I would need to hear that they were extremely dissatisfied with their current quality of life to consider an experimental therapy like this. And although I didn't mention it before, I'll just mention quickly that the other thing that we have to keep in mind is that it's possible that the recipient of a xenotransplant, of a pig transplant, might need access to special medications, and I would want assurance that they would get access to those medications as long as need be. And I'll stop there. Thank you.
Thank you, Dr. Reese, for that amazing presentation. So now we will open it up to a question and answer session. So in order to make this systematic, I would like for everybody to enter your questions into the question and answer box. It should be, if you're on a desktop computer, type them in at the bottom of the screen. You should be able to click Q&A. And then the Dr. Reese and Dr. Kumar will answer your questions until the end of the webinar. If you have further questions, please feel free to email me directly. So would you guys like to just read the questions from the question box and tackle them?
Sure. It says, is there a concern that recipients' insurance will not cover the cost of medications after transplant? I think that is a concern. I think that's the kind of thing that really would need to be ironed out before the transplant. And I think that, if there's an expectation that some medicines would be covered by insurance, that's the kind of thing that we would probably try to address with pre-authorization. My experience with some other studies is that you never really know what an insurance company is going to say until you ask them. Sometimes the answers can be unpredictable. So I think some medicines would probably come from the study team, but others might need to come from the insurance. I don't know what you think, Vanita.
Agree with you, Peter. Well, two things to consider. One is doing these xenotransplants under the cover of what is already being used in human-to-human transplants today. That would be ideal. That's a group of scientists are working towards that effort of just using what's already available. So the same rules would then apply for post-transplant coverage. I see Glenda Roberts' question about whether Medicare would cover drugs for experimental xenotransplantation. So for the next steps, because we are advocating for it to be under the prospect of a clinical trial and not just available for public consumption at the present time, for the next few patients, if a clinical trial comes to fruition, the if there's use of experimental drugs, it would have to be part of the study design and be paid for through the study, as I doubt at the present time Medicare would cover experimental medication, just like it wouldn't cover experimental medication for anything else we do in medicine when we're studying it. And Peter, what do you think about that?
Yeah, I agree with you. I think that insurance coverage for drugs when there's an experimental therapy, my experience, at least with the hepatitis C trials, is that we were occasionally surprised. But hopefully with kind of a pre-authorization type of situation, we could get some feedback from Medicare or in a private insurance company before the transplant went ahead.
Super, yeah. Brianna Bardham has a question. Do pigs have two kidneys like humans do? Yes, Brianna, pigs, at least the ones that we've all encountered so far, just like humans, overwhelmingly have had two kidneys. There's an occasional human that's either born with one, has one that lost along the way, or not developed fully. Probably is true for pigs, but for the most part, pigs do have two kidneys.
I see one question we can answer, which is, would a xenotransplant be an option for individuals who've had previous transplants? I think the answer is maybe. I think for any individual who's had prior transplants, we should subject them to the same assessment in terms of, is their health robust enough to go through a transplant? And certainly for some patients, particularly young patients, the answer would be yes. But if a patient's had multiple transplants and has comorbidities from it, cancers or other health problems, then that could rule them out. But I don't think having a prior transplant would rule them out. The other thing about people who've had a prior transplant is their immune system is sometimes more sensitive to how it responds to antigens. So they might be more likely to react to the pig tissue, possible. But we have ways of testing that, and I think they'd be presumably tested in a pretty similar way. So I don't think it would rule them out, but they might. But we would try to work through their medical situation systematically.
Agree, yeah. When we consider patients who've had prior transplants for even human transplant, the couple of big considerations are, is there space to put that new kidney in, right? Surgically, can you hook the kidneys up? And so that assessment would be true also for the pig xeno kidneys. We have found from doing the decedent experiments that we can do the surgery very similar to the human-to-human surgical transplant. So that's one thing. And the other thing is in patients who've been transplanted before, they can form a lot of antibodies, exactly like Dr. Reese alluded to. And so that does reduce the pool of acceptable human organs for that group of people who've had all these antibodies. And the question is, will these patients then also have some cross-reactivity to the pig antibodies? And at least in preliminary studies, it looks like a segment of these patients will. But for that reason, we now have a test developed to match the pig kidney to the human, just like we would do human-to-human matching before we even move towards transplantation. So that's another tool we have.
I see that Melissa asked, what percentage of your patient population do you think would benefit? And I think the answer is we really have no idea. And we have to do these experimental studies to find out. When my colleagues and I have tried to think about who's reasonable to consider for the studies, we really tried to think about the profile of people who are very unhappy with their other options, that are open to a risky therapy, that are resilient enough to make it through it. But if we enrolled someone in these studies, I don't think that we would say you're likely to benefit. I would say we're here to find out if there's a benefit. And participating in research studies, patients are often hoping to get a benefit, and sometimes they do, and we would hope that they would. But the main purpose of a research study is to generate knowledge. And many times we are deeply uncertain about whether patients will benefit or be harmed by it. And that would be true here.
Absolutely, yeah. I wonder if we should tackle this question now, Dr. Rees, but I'm going to read it. The first one in queue here is assuming limited resources so that not all increased kidney supply approaches could be maximally pursued. How would you rank increasing deceased living and pig transplant? That's a pretty important question, difficult to answer, but I will take a stab at it. I think if we had enough deceased donor human organs, we may not be looking as much to living donor organs, knowing that the best organ that today a human can get, at least for a kidney, is a living donor kidney organ, just for multiple reasons, which I won't go into here. But if you had enough deceased donors, maybe we wouldn't even need living or xenotransplantation. But since probably the last 5 decades of trying, we simply don't have enough deceased donor organs. We could continue to make efforts. We have made efforts. The number of organ donations has gone up. The recipients waiting has also gone up as the US population is aging, living longer. Kidney disease is being picked up early enough where we can mitigate death. People are living with kidney disease requiring more transplants. So continued efforts in the deceased donor space, absolutely important. I would rank that one. Continued efforts in living donor space, very important. And I would actually say that while I say pig xeno number 3, if this works, the potential for it to explode and actually really fill the gap between the people waiting for a transplant and getting a transplant in a meaningful way, the potential for pig xenotransplant to me seems to be the greatest, and the next few decades will tell us. But Peter, how would you answer that question?
No, I like the way you answered it.
Okay.
I see Catherine Avery asked a question. Since African-American, well, she just moved. So African-American, Spanish, Latino, Native, Indian, and Asia Pacific populations are affected the most by CKD and kidney failure. Why would a 50 to 59-year-old patient be asked to participate over those who are affected the most? I don't think that anything that was said today would suggest preferring a person of one race over any other. The example that I was giving there was just an arbitrary example. If you look at the paper, you'll see that we identified people from many different backgrounds who are more likely to die than get a kidney transplant. But I am glad that you asked the question, because I do think that equity is a huge issue in kidney disease. And I actually have a paper coming out soon where we were just trying to make the case that there's been so much inequity, racial disparities with access to human kidney transplant, that maybe xenotransplant is a really wonderful opportunity to kind of take a fresh look at what sort of equity solutions are there. And that would certainly apply to participation in clinical trials. I would want what I think would be really great is for the xenotransplant companies to partner with centers with demonstrated evidence that they well serve the needs of Black and minority communities, and hopefully have large minority populations and trusted relationships, and maybe advisory boards with minoritized patients who really contribute in a meaningful way to the direction that the center takes. So I think equity is a really important issue, and I think it should be front and center as these trials are developed.
Beautiful, I love that. Melissa Hardwick asks, what medications would be required after a xenotransplant? Would there be more than what is required for allotransplant? They can be harsh. And yes, absolutely, Melissa. When we listen to our patients, the number one thing they tell us is the currently used typical cocktail of tacrolimus, mycophenolate, and prednisone, to take it day in, day out, and to take it for years on end is not only is it difficult to do that, but the side effects and to live with those side effects, we can only imagine how tough that would be. those currently the way Xeno is being developed, it's developed in different ways at different centers, but using the standard immunosuppression is a thrust that we definitely have when we're partnering with our Xeno companies and doing these decedent trials, and we're using the standard immunosuppression. It's looking like we may need to do some additional medications, but medications that are already in use, not experimental medicines, but that's a very important question, and time will tell where we'll land with the holy grail of immunosuppression. Melissa Sanchez says, if the xenotransplant fails, would the kidney be removed from the patient or left in as in a human kidney transplant? The current thought is if the xenotransplant fails, or if the xenotransplant for some reason is causing adverse impact in the recipient, then at least the working plan for most people who are working in this space is to remove the xenotransplant. Earl Collier says, why are human kidneys removed in xenotransplant versus human transplant? And in the decedent models, the decision to remove the human kidneys was done to show that in the absence of original kidneys, because the decedents could have functioning kidneys and just not be able to donate for other reasons. So in the absence of their own kidneys, can the xeno kidney provide the same function that we require of a human transplant? But when we move to clinical trials, the plan is not to remove the original human kidneys, just like with human transplantation. There might be benefits to leaving the human kidneys in terms of some hormonal production from the original kidneys. Peter, are you seeing other questions you want to tackle?
I saw this question from Sandy Dotson about being unsatisfied with dialysis. We didn't answer that one yet, did we?
No, we haven't. Yeah.
Okay. So Sandy was saying, when Dr. Reese was stating the specifics for consideration of xenotransplant, he stated that they must be completely dissatisfied with their quality of life on dialysis. Suppose they've accepted being on dialysis as a way of life, not completely dissatisfied, but would like to advance their quality of life. You know, I think it's a judgment call. My general take was that if someone is going to embark on a xenotransplant, then I would want them to have kind of a restless dissatisfaction with the life that they're living that would motivate them to feel like it's worth it to take a risk that is of unknown size. We can't even say with any confidence how likely it is they would reject the kidney in the first 3 months. And so I guess what I would say is that I would want a patient to come to me and say, I need to do something about my situation. I don't see myself thriving. I don't see myself, being content in this situation. I have to do something. I think that that's the kind of person who would be reasonable to talk about a xenotransplant trial. And, and it's not just the risk of rejection, the risk of infection, the unknown therapeutics, the possibility of being in the public eye. It's all those things. Now, I have some experience with this from leading some of the trials with hepatitis C. And I think Benita may have participated in some of the trials where, there have been other experimental facets to them. And I'll talk to people on dialysis, and some of them say, I'm on peritoneal dialysis, going pretty good. Not really feeling like I need to get involved in your study because I'm happy with how things are going. So when I meet those patients, I don't try to talk them out of it. I just say, you seem good, stay with what you have. But sometimes those patients can come back 2 years later and say, boy, I was fine then, but I'm not fine now. I had an infection, I had a heart attack, I was on dialysis, the person 2 chairs down coded. That was very uncomfortable for me. I need to do something different now. So I feel like that's the kind of urgency, the sense of urgency that I would be looking for. Not everyone may agree with me, but that's how I put it together.
That lands and resonates with me too, Peter. Yeah. John Clouston asks, is there any evidence at this point that says patient's blood type might influence ability to accept xenotransplant? John, that's a really good question. And currently the way the gene-edited pigs are being developed, the pigs are going to be universal donors, so we don't have to match them for blood type. They're all blood type O, essentially. So that's one less worry about matching for especially a disadvantaged blood types for organ transplantation. Yeah.
Okay, we are actually at the 4 o'clock mark. Do you want to answer one more question and then we can call it for the day?
You know, there were a few questions about age, and maybe this is just a good opportunity to address that. At this point, I don't think that there are any age criteria for xenotransplant. I don't think any trials with specific inclusion criteria have been accepted or are out in the public eye. So I don't think there's any age limits that we know of right now. One of my slides, I just gave an example of a person aged 50 to 59, but it really was just meant to be an example. It's not a person that there aren't any age limits for xenotransplant right now. We don't know who's going to be allowed to come into a trial. But that's what we're here collectively as a community to have input on.
Well, thank you, both of you, Dr. Reese and Dr. Kumar, for your time today. Everybody that has asked questions, I am able to save these and I can contact the chairs for their answers and I will send them out through email. If you have any additional questions, please contact me directly, heather.murphy@kidney.org. Thank you all for your time. And is there anything else that either of you would like to say?
I really want to appreciate the time that all our participants are giving to this project and to move things forward here, to want to understand, learn, and help impact the field, and to NKF for putting their efforts behind this. And I learned a lot listening to you, Dr. Reese. Always a pleasure.
Likewise.
Yeah.
Wonderful. Well, thank you, everyone. Have a wonderful evening.